Cytokines include several subtypes, such as interleukins, interferons, tumor necrosis factors, and growth factors, among other things. Interleukins are of specific importance because they are a specific type of cytokine. Interleukins are numbered, and each of their numbers represents a very specific function that that interleukin does. The term interleukin comes from inter, meaning between, and leukin, meaning white blood cell. And that's why interleukins are mainly produced by and act on white blood cells. Cytokines, interleukins, are like communication between white blood cells. These proteins can recruit more immune cells, like neutrophils, to leave the bloodstream and come to an infected area. Some interleukins tell neutrophils to stop their processes so that tissue repair can begin, kind of like regulating the immune response. This diagram shows you how these types of T cells in the left-hand column, they all secrete different types of interleukins. The interleukins have a variety of effector cells like basophils, neutrophils, dendritic cells, and they can recruit them to come to the injured area and help with amplifying the immune response, kind of creating something like a positive feedback. So just one of these T cells can secrete an unlimited number of interleukins, really, and can recruit thousands of these effector cells. Interferons, which sound like interleukins but are markedly different. Interferons are proteins secreted by infected cells, virally infected cells. And they're kind of like a last ditch effort to save any adjacent uninfected cells from experiencing the viral attack. So interferons diffuse into adjacent cells and the interstitial fluid, and they cause the still healthy cell to synthesize proteins that try to stop viral protein synthesis and proteins that try to degrade the viral RNA. Interferons are chemicals secreted by various cells, healthy as well as immune cells. And they play a big role in signaling to other cells that they're virally infected. Alpha interferons are produced by virus infected cells and are sent out as that last alarm signal. Beta interferons, secreted by fibrocytes and connective tissue cells, they regulate inflammation and they stop inflammation and start the repair process, particularly in autoimmune conditions rather than infections. Gamma interferons secreted by damaged skin cells stimulate macrophages in the dermis, supporting the immune response. It is these interferons that are oversecreted in diseases like psoriasis, where the skin cells believe that they are damaged, secreting the interferons and increasing the activity of macrophages in the dermis. I really I really dislike the complement system. Not because it's useless, it's very useful. I just dislike it because every time I get a new textbook, there's so much more we know about the complement system, and I have to change everything. That's okay, that's all right. The complement system, it's a set of at least like 20 proteins, plasma proteins that circulate in the blood, and they're inactive as they circulate. So they have four main functions. Number one, they set off inflammation. They can also stop it too. They can cause cell lysis, so they can actually insert a hole in the cell membrane of a pathogen. They can attract phagocytes, macrophages, and they can make the macrophages more hungry. Complement can be activated by antibodies, or they can be activated when certain complement factors interact with microbial cells. Complement proteins can be activated in more than one way. The classical way is really fast and really fastest of the two, and it's antibody based. So basically, antibodies that are stuck to a pathogen attract the complement proteins. The alternative pathway is much slower and tends to be used for parasitic infections and viruses. It doesn't matter though. The result of both pathways includes enhancing inflammation, initiating cell lysis, attracting phagocytes to the area, and promoting phagocytosis. Complement proteins, they mediate inflammation by stimulating the release of histamine from mast cells and basophils. And we know that these two substances are powerful vasodilators and increase the permeability of capillaries. The complement system can bind to the antibodies that are fixed or embedded into a target cell membrane, so their own. A group of complement proteins called the membrane attack complex, or MAC for short, are inserted into the cell membrane. And the membrane attacks complex makes a pore big enough in the target cell, and it just basically induces osmosis, and the cell shrivels and dies. Complement proteins are used to attract phagocytes to an area or to a specific pathogen, and this is called opsonization. The root word for opsonization means condiment. Just as ketchup or whatever your chosen dipping sauce is, complement can make a pathogen just more tasty for macrophages. A product of the complement activation is something called the C-reactive protein, or CRP. Many times, if cancer or inflammatory conditions are suspected, you might get the CRP level in your blood measured. So CRP, it's a protein produced by the liver in specific response to cytokines, interleukins, and other things that cause inflammation. Um, it does have a useful clinical application. It can be supportive of things, but it usually is used to rule things out rather than confirm them, because so many things can cause inflammation. But C-reactive protein is also used by oncologists to see if a patient is responding to treatment.